Hi everyone. But by the way, do you hear me online? Is this working? Like we tested this before, but I hope it's cool. Thanks a lot. Thanks a lot for it. Hey. Yeah. So welcome to this course. It's actually a great pleasure, and we were looking forward to this course like since several weeks, and we tried to prepare it and to make it very nice and fun. Yeah? So I hope you will enjoy it, and please give us our feedback. So this is an experiment in some ways. Yeah, we changed a lot from last year. The biggest change being actually there is an online audience and an in-presence audience. Yeah? We try to make it very nice for everyone, but like just give us hints like what we can improve whenever there is something to improve. Yeah? And perhaps the big, yeah, let's start with first things first. Yeah. So who is actually giving this course? So this is Thomas. I don't know. Do you want to very quickly be visible in the camera that everybody sees us? And Mariapia, if you perhaps step here also. So we three actually prepared this. Yeah, hi everyone. So we three actually prepared this. So Mariapia, she's a PhD student here. With us, working on bioengineering, and perhaps there will be a time later where you can talk a bit about this, in principle, what you're doing. And Thomas and myself, we are working here at ENS Paris-Saclay, so we are in the Greater Parisian area. And yeah, Thomas is a professor here, and we're a researcher here. Yeah? Okay, thanks a lot. Yeah. So very quickly, like I want to make this, of course, very fast for you. So our principal way to communicate will be in the Discord forum. Like you all should have received invites, I guess, for this Discord. Yeah. If not, yeah.
Not yet.
Okay, not yet. Okay, sorry. Yeah.
So everybody online should have.
Okay. Yeah.
But you guys haven't.
Yeah, probably. Okay. Yeah. Then please, afterwards, like in the problem session, I think we will just figure this out with you. But you will have, you will receive Discord invites, and then because then we have everything together in one big forum, and we can change there. Yeah? Okay. So let me perhaps go very quickly to the modalities of this course. So this is not a classical lecture. This is actually a flipped classroom lecture. But we are not completely flipping it. We are flipping it by 90%. So our principal idea would have been, we give you, like, essentially you do a pre-reading of the lecture material that is already online. I mean, it will change probably during the next weeks a little bit, but like everything that you should know for the next lecture is already online. Yeah. So even if it changes, this is completely fine. We will just add some smaller details. And in the first hour, one of us is going to present a little bit like a very quick summary, but this is not thought of as a lecture part. So from this very quick summary, this will be too short to really get and digest the big material. Yeah? It's really just as a reminder for you, a refresher for you. And we will in particular get into questions that you post in the forum. Yeah? So we have received several questions, and we will, of course, when it's time, go through these questions. So yeah, exactly. So actually, this brings me to another point. Like, please post questions whenever you want something explained, or you want more details on something, or some really like it's in part it's like a you can announce your wish list here. Yeah. So and we will try to accommodate this. Okay, for the prerequisites, I mean you probably come from very different backgrounds. So we saw actually in the registrations, like we got biologists, we got microbiologists.
Recording in progress.
Yep. Is it? Okay, no, sure, perfect. We have people from AI, from computer science, classical computer science, like from quantum physics. We are a very diverse set of expertises. So some parts might be boring for you because you know them by heart. Some other parts might not be. So we try to focus or to bring everybody to the basics like as quickly as possible. So in part this will be actually this lecture here, but probably also the other lectures a bit. And there is no real background that we say you have to have this. I mean, it will be sometimes a bit mathy and but you don't worry too much, even if, I don't know, some integrals will freak you off. Like, but this, we will go over them much more in a simulation perspective, so don't worry too much about it. And you should perhaps bring some Python coding skills, yeah. But also don't worry too much. It's not really a Python coding class, yeah. It's really we use very basic principles of coding, yeah. Yeah, and perhaps that finishes almost the prerequisites, just with some very nice biography. Yeah, you can find here some books we really like a lot, and feel free to check them out and look into it. Oh, one more thing perhaps to say to the lecture when I start this, talk about this. This lecture has already, I think it was it 1 or 2 years ago, like when we have forked them from a Caltech lecture. So when we started this lecture, we were looking around like, what do other people do? And we really liked a lot the Caltech lecture. So this is actually a fork from here. In the meantime, it has developed a bit its own spirit, but it's, you will recognize a lot from this very nice lecture by the colleagues at Caltech. Yeah? Okay. So this is just, since we have been asked this in the forum, so those who are already very familiar with coding, you can close your ears here, but essentially people have asked like, how do I run the code that we have here? I mean, you can download it on the website, yeah, but the way I run it, for example, is actually in VS Code. So this is freely available, yeah. I think on all platforms. I run it on the Mac, but I think it works on all platforms. Yeah? And perhaps be careful to create your own virtual environment, because we will install a bunch of libraries. Yeah? So I mean, you can fully go cloud also with Google Colab or whatever you like. Yeah? That works also pretty nice. But don't overthink it. Don't overdo it. It's pretty simple, small code that will not run for very long. So you don't need an incredibly cool setup for this. Yeah? But perhaps make this setup very in the beginning, because I think it's very nice to play with the code while we are talking. Yeah? So I would suggest that if you do not have this already running in your Jupyter notebook, do that perhaps in the next minutes, or at latest in the problem session you should have let it running. Yeah. Okay. Yeah. And by the way, if you have any difficulties with this, yeah, because sometimes this is a bit tricky to do, just ask for help in the forum. And in general, like if somebody sees a question that you can answer, please help us answering them. Yeah. And if not, of course, we jump in and try to fix things as good as we can. Okay. All right, so yeah, this is really just some. I'm getting slow here in the beginning, but we're getting, of course, faster than later on. This is really just how to install stuff. Yeah, like for example, when something doesn't run here, don't worry. Like the way to do this in Jupyter is with this %pip install, for example, NumPy. If it complains about NumPy not running, you just uncomment this and you install it. And this is the same for all libraries. You just with this magic commands that start with a %, you just install the libraries. So. If you go with pip. Okay, so I think that's the prerequisites. And that brings me to the introduction of the cells. So what are we going to do? Like the course is called Computational Bioengineering. So we try to bring together computer science, which is actually our background. We come from computer science and computer engineering, yeah, and biology, in particular microbiology. And the things we were super interested in, and we asked ourselves actually now almost 10 years ago, yeah, it's like, can we actually use the tools that computer science has developed a little bit on microbiology? And the quick answer is, of course, like there is a bunch of research going on. I mean, I mean, bioinformatics is as old probably as computer science, yeah? But like to use them in a more direct way, let's say, not just like helping it like bioinformatics way, but like really more directly engineering these things in a computer science way, yeah. And this is the point. This course should give you the tools a bit to do that. I mean, we are far from claiming that this is after this course you can do that pretty well. I mean, because it's a very developing area, a super cool area. But like we try to tell you a bit what we know about the systems. So let's start with the cells. I've been asked if some of this material actually holds for mammalian cells. Yeah. And the answer is yes, yeah, in principle yes. The examples we will be giving, they are not from mammalian cells, they're actually from a bacteria called Escherichia coli, E. coli, yeah. And the pure reason being that we work with it in our lab. Yeah, so we are here in a computer science lab, but we have a little wet lab where we can run real experiments. And we did not yet have the time or energy to look into mammalian cells. But we have some ideas to do that later, but not yet. But actually, we might have some specialists, or I guess we will have specialists in the forum. So if you're a specialist in mammalian cells, please give us your views. Yeah? Like we're super interested to have your opinions on this also. Okay, so which cells are we looking at? So we are looking at an E. coli, and in a very simplifying way, let me show you this picture here. This is actually a microscopic picture of an E. coli. Yeah? So this cell is roughly 2 micrometers long and half a micrometer thick, yeah? And it's in a rod shape. So you see that here. These 2 micrometers, of course, this changes. You see that here versus here, whether it's just shortly before duplicating or just has duplicated. Yeah, of course, this is very varying. Yeah? And this is like a cut, a systematic cut. Like you see actually this section here of this E. coli here. And what you see actually is that here is the outside of the world. Here it has this lipopolysaccharide that are some small hairs going away from its surface. Yeah? Here's the outer membrane, so this is the first barrier from the outside to the inside. Then there is a periplasmic space that is actually a bit in between space. Yeah? We are not yet completely in the cell. And then there is the inner membrane, which is now really the border to the inside, the cytosol of the cell. Yeah? And since it's a bacterial cell, more or less, everything is mixed up in this inside. Yeah? There is no classical compartments like in mammalian cells. Yeah? But of course, like this is only half the truth. Yeah? Of course, there is a structure here, a spatial structure, but we will not go into details here. Just for the first picture, this is completely enough for us. Yeah? Yeah, and perhaps one thing to say is, so how does stuff like nutrients get from the inside, from the outside into the inside, or toxic stuff from the inside to the outside? This is actually these little things here. This can be actually just by the diffusion of gradients, yeah, because there is a difference in concentration, and it can be actively transported. So the cell can actively, with taking energy, transport certain stuff inside and outside. And you see here actually like this. Okay, this is a very thin layer, yeah, and you have very interesting proteins stick into from each side, or through, or even cross membrane, yeah, that are inside this and that have certain functions. Yeah. So I think what is really nice to see, I think everybody should see this video once. Yeah? I was completely fascinated when I saw it first, is this little video. And this is, okay, you don't see this here unfortunately very well in the screen, but you can click it through in the Jupyter notebook. Yeah? This is actually, I think it's 15 milliseconds of an inside of an E. coli. So this is like, you go into this E. coli and you put on your camera and you look around how it's looking like. Yeah? Because this rectifies completely how it's often drawn. Often it's drawn like as this big thing with this genome here and this ribosome here and stuff like here. It's completely crowded space. I mean, this is like really in the prime time taking the RARB. Like you're squeezed like this and you have to think like all chemistry is happening in this crowded space here. Yeah? So if something here is reacting with here, I mean, they have to squeeze through to find each other. Okay, let me close this for the moment. I think this is another pretty nice picture that you might have looked at already. It shows the, at the moment, known pathways, so most metabolic pathways, yeah, of the E. coli, like structurally, and all these reactions. So like each small line here is a small reaction, yeah, and these pathways are actually these lines of bubbles and lines, yeah. So reactants and reactions. And we see that there is a lot of things going on here. And I mean, this is only the known ones. Yeah? For example, you see here very short ones. Probably there is connections here. I mean, probably it's not just this guy reacting with this guy and then that's it, standing for its own. Yeah, but they are just not known yet. Yeah. So, and there is of course more to come. This picture is growing every year. Yeah? So this is pretty impressive. There's interactive versions, so I encourage you to play around with this. Yeah.
Every single dot is a what?
Yeah, it could be molecules. It could be tetramers of molecules. Like it's something, some chemical or microbiological species, yeah, that we will call it, that reacts with something else. Yeah, yeah. But it could be one molecule. It could be one complex of molecules. Different things. Yeah. Yeah.
Kind of like automata. They look like states.
It is actually I mean, if you can view the state space, but I would say the state space, of course it depends on your modeling, but the state space could be considered even bigger, yeah? Because, I mean, imagine, for example, this pathway being in this state, and this pathway being in this state, and this pathway in this. So you could think of the Cartesian product of all this, like this completely an explosion of everything, yeah? But if it's kind of parallel state machines, if you want, yeah. Mm-hmm. Completely, yeah. Yeah, and here is like, perhaps this answers a bit your question. Like this is one zoom into one of these reactions. Yeah? So you see, I animate this is very graphically very nicely. So this would be one bubble. This is another bubble, and this is another bubble in the picture. Yeah. Yeah.
Just to be sure, a pathway is a sequence of reaction.
Yeah, completely. Yes. Yeah. So we will actually good questions because now we go actually to pathways. So thanks a lot for this nice transition. So perhaps I start with circuits, yeah, and then I go to pathways, yeah. So some of you might come actually from an electrical engineering or computer science background, yeah, and so you probably know what is a circuit, or you know it from school. Like in principle, like what is a circuit? Like speaking of a circuit, I mean just really the in silicon circuits, the electrical engineering circuits, the classical circuits, yeah. So typically they have inputs, right? Where you give some signals, and these are typically encoded, for example, in binary 0 or 1 or something, yeah, as voltage states. And it has kind of outputs where you're expecting or a certain type of behavior. And then it has some internal components that are transforming the signals. Yeah? And we will actually see that such circuits, yeah, for example, use such components, yeah, right? Like you have the simpler components on the left side. These are passive components like a capacitor, an inductor, a resistance or something, yeah. They are kind of connecting two nodes in this circuit. For example, a resistor, you can put between two different nets in your circuit, yeah? And then it imposes a certain relation between these 2 points. But it's very important to say that actually a resistor doesn't have an input or an output. Yeah? So I just told you that the circuit has an input and has outputs, yeah? And the resistor doesn't have an input or output. Yeah? Or a diode doesn't have an input or output. This doesn't have an input or output. Yeah? So actually, you're creating something directed from inputs to outputs with something that is a priori undirected. Yeah? That doesn't have dedicated inputs outputs. You should much more think of it like constraining the charge on these two nets. Yeah? So it's kind of a constraint that relates the voltages and the current in these systems that you connect to it. Yeah? And I think this is something very important because when you look at circuits, of course you would say, my current is going into this resistor with this input current, and then and I get this output current, which is the same, for example, and so. But it is actually us that think of it in a directed way, or that use it in a directed way. Yeah? And this is even more extreme if you think of digital circuits. I mean, they clearly have now inputs and outputs, yeah? But a priori, as the physical components, they also don't have inputs and outputs. Yeah? I mean, we use it just in a way that we view these ports as inputs and this port as output. Yeah? But a priori, it's again something, only a constraint, a set of constraints that's working on these nets. Yeah? And this is kind of an abstraction that helps us to have inputs and outputs. Yeah? And the same is like for pathways. You can view this. You can view all these things, yeah, as being directed in the way that it has this input species here, and it's being transformed, and it's being transformed into this output species, yeah. But a priori, chemistry is undirected. I mean, this is just our interpretation and our way to have a simplistic view on these things, yeah. But yeah, perhaps there's a bit from the commonalities about these pathways and circuits. Yeah? And they have a lot of commonalities. So for example, in this lecture, we will always use circuit and pathway in the same way. Yeah? So for us, a pathway is just a biological circuit. Yeah? And there is no big difference a priori. Yeah? I mean, the rules are very different, of course, yeah? How things are transformed. But the abstraction principles are the same, essentially. Yeah? And to make this point a bit more fundamental, what we did is actually we looked at some example electrical circuits. Just let me check the time. Okay, I'm good in time still. Yeah? So some example electrical circuits, and the purpose of this whole exercise was not from you to make experts in analyzing such circuits. Yeah. I mean, this takes a lot of practicing and so on, yeah, to get very quick in this, and to solve the ODEs of this. It was much more to see how this has commonalities with later on what we see as biological circuits. Yeah? And the commonalities will be, and this is a bit, it's cool and dangerous at the same side. Yeah? At the same point, like, they have a lot of commonalities, and at the same point, we should be very careful in not taking this picture too serious. Like, for example, if you did the pre-reading, you will have found out that this circuit here, which is our most simple genetic circuit, yeah, I will say a few words about it later on, yeah, has a lot in common with this electrical circuit, yeah. Yeah? So essentially, if you map some of the parameters of this to the other, it has the same behavior. Yeah? And that's cool and dangerous at the same time. So it's cool because you see, like, there's common principles between both types of circuits. Yeah? It's dangerous because don't be trapped in a way that you try to model everything with such circuits. Yeah? I mean, I think people did this in the beginning. And I think it's kind of a dead end because it's not the components. I mean, we might have things that act a bit like a resistance and so on, but don't take it too serious. Like they need their own components and their own language, of course. Okay. Perhaps a few words about this circuit. Did everybody, like, including the online audience, and of course here, yeah, did everybody understand, like, this circuit here? Like what it does and what it is and so on? Or do I need to say some words about it? Or was it completely clear? Okay. Just take a sip of water in the meantime, like, while you look at it. Okay, so I see a lot of thumbs up. That's great. So I will not go into. Or there was no one else who. Yeah. So I think then we can go on with this circuit. So thanks a lot for the feedback. Perhaps a few words to say to this circuit that is important when you parameterize it later. Yeah. So we have this reaction that is actually degrading or diluting stuff. Yeah? And of course, this depends a lot what this gamma will be. This depends a lot on this product. Like, is this an mRNA? Is this a protein? Where is this mRNA? Are we in a mammalian cell or are we in a bacterial cell and so on? Yeah? But the interesting thing is, like, for bacterial cells, most of the time, I don't say always, but most of the time, this is actually, the big thing is actually dilution during growth. Yeah? For example, like most of the time you will look into your circuits when they are in the perfect exponential growth phase. Yeah? So they duplicate as fast as they can, roughly every 20 minutes. Yeah? So, but by duplicating every 20 minutes, you are essentially halving the concentration of whatever was in there every 20 minutes. So this is a big part of the gamma. Yeah? And this is probably what's really determining this gamma, yeah, most of the time. If you need to start, then this is probably a good start. But I think this is nice to say. And the other nice thing to say is we were a bit sloppy when we painted, or actually I don't know if we painted or stole this picture, but like when we showed this picture, we were a bit sloppy, yeah, because you see here two different types of errors, kind of. Yeah? You see a transformation error and a generation error, let's call it. Because this error here doesn't mean like that the gene X is transformed into a protein X. Of course not. I mean, the gene X produces, it's still there afterwards. Yeah? And this error means that this is really transformed into nothing. Yeah? So it's not producing nothing. Otherwise, we would not need to paint it here. Yeah? So we sometimes use these a bit sloppy languages in pictures, but be careful, of course, when you model it. Yeah? It's two different types of reactions, whether the reactant is still there afterwards or not. And the other thing, you might have seen this gamma X. So this is a bit strange, because why do we have a beta here and here a gamma X? Does somebody know? Yeah?
The rate of degradation is
yeah.
proportional to the number of proteins.
Completely, yeah, exactly. So I'm just repeating it for the microphone. So as you said correctly, like the rate of degradation is proportional to the number of proteins that are already here. And this is intuitively true, yeah, that it's linear. We can actually prove this. It's a very simple proof, because you assume here an exponential decay, yeah, and if you have X exponential decays happening in parallel, you get actually the decay by -1 for the first of this happening, yeah, the minimum of them happening, is actually the sum of them, so is product, so it would be gamma times X, yeah. But you will see the math perhaps later, I think. Yeah. Okay. Let me just check one thing. Yeah, I have added some side notes, because this is something like we kept all the time repeatedly making errors ourselves, and together with students, about units. Yeah? I mean, here I was not very careful about units. I mean, here you see just the ODE, this one. But it's very important, and it's a very nice assertion to check if your modeling is going right. If you please always check your units, yeah, and even make your code to check the units. That is the best. Yeah? And it's not that trivial, because for X you have actually several reasonable choices, and all can be argued to be actually okay. Yeah, you just have to stick to one of these choices. Yeah? and I've written down this is in these side note units, but just to give you the glimpse of it very quickly. Yeah. Of course, like, let's say the most natural we are going to use all the time is actually X being a concentration. And again, for concentration, you have again two choices, yeah. And we are looking at concentration as counts per volume, yeah? Most of the time, if it's not otherwise noted. Yeah? For example, that could be, I don't know, 5 mRNAs per femtoliter, femtoliter being roughly the volume of an E. coli. Yeah? And then once you fix this, all the other stuff is fixed. Yeah, what is the units of gamma and so on. I mean, you you just make the unit game where. What else could you have chosen for x? You could have chosen, for example, molar concentrations. You could say how many moles per liter this is. Would have been perfectly fine. Just since it's normally something like 5 mRNAs or something, it's a bit weird to write molar concentrations because you don't have so much a feeling if you look at it. I mean, except if you're very good, but if you look at it to recognize something is 5 or 70 from the molar concentration is not so easy. And the last, and we will actually most of the time not do that, but sometimes it is interesting to do that. You could, is there something? Yeah. You could see an x as actually being an expected number. So we are not looking into concentrations anymore. We are fixing the volume. Yeah. Let's say we have fixed the thing that we are in this 1 femtoliter. We don't care, and we say x is the expected number of mRNAs we are going to see. So it's a number. Yeah? And again, then all the other gammas and so on, betas have different units. Just be super careful whatever you choose to make it explicit and carry it through. Yeah? Okay, so how did we get this ODE? I mean, this ODE dropped somehow out of nothing. Yeah. There is a more systematic way to get it. And this is actually from something that looks like this kind of reactions. Yeah. Here they are made explicit. So by the way, yeah. So this reaction is clear, right? We are having a degradation reaction. We are degrading a protein into nothing. Yeah. This reaction is a bit less clear from the picture we have just shown because I just showed you that it's actually a gene producing some X. Yeah? Just for the simplicity of the model, to have only one state variable, we have just removed the gene. So we say it's nothing that creates X because the number of genes is always constant in our simple model. Yeah? This is why we have just removed it from the model. But you could add it, of course, if you want to. And I think it's actually the next lecture that we will see a really systematic way how to go from these reactions to the ODEs. For the moment, I just tell you that you can do that. Yeah? And why do we tell you that you can do that? Because we thought it's actually nice at the first lecture. Even originally we had this quite far back in the lectures, only after some lectures. So we have actually, like with a former PhD student, Fabrizio, who is now a postdoc in the US, and Gayatri, who's also in the US, yeah? Right now, like we have created a tool that can simulate such reactions in a nice way. Yeah? I mean, here we are really using it in a very simple way for the moment, yeah? Just for these two reactions. Yeah? You can just, I mean, you almost see these reactions here. 0 goes into X, and X goes into 0 with this constants. Yeah? But it's very useful for you probably during the problem session already to prototype stuff. Yeah? You can just play around with it, and even if you don't yet completely, didn't discuss how it's doing that and what it's solving actually, you can just use it for the moment. Yeah, and if you do that actually, so this was created with MobSpy, you see actually it has this behavior that you expect from the ODE. Right? Yeah, here's the same MobSpy code. I was claiming that please use units, and then I was not using units. This was just to show you a simple code, but here is the kind of code that would be nice if you write that yourself, because like here it really checks all the units for you, also this MobSpy library. So that's quite a nice thing. And of course you get the same output. Okay. Let me just give me a second before we start with steady states. Huh? Okay. So, of course, you can either analytically, yeah, like in our case, or numerically simulate or solve these ODEs. Yeah? But sometimes actually this is not at all what's needed to understand it. So it's often a very good idea just to look at the steady state. So, and what is the steady state? I mean, a priori, it's not necessary to exist because your solution could be an oscillatory, or it could just go to infinity or something. Yeah. I mean, for genetic circuits to make sense, most of the time it's either oscillating or it's actually going to some limit. Yeah. And in our case, actually, it goes to some limit. And I don't know if you remember from your math classes, but you can very easily get these steady states once you're sure they exist. And in this case, we guarantee you that they exist. Yeah. But just setting all these derivatives to 0 and just solving these equations. And here we very simply see that it's actually one rate parameter over the other rate parameter. Yeah? So the production rate parameter over the degradation parameter. And if you play around with increasing the production and or increasing the degradation, you see that the limits make sense of this. Of course, if you go, for example, with gamma, the degradation to infinity, the steady state should go to 0, and it really does that. Yeah? So that's nice sanity checks that you can always do. Okay, of course, like you can go arbitrarily complex with these models. Yeah, I mean, even though that it's a super simple circuit, I mean, just a gene expressing a protein. Yeah, you can already go crazy with such a simple model. Yeah, I mean, here we show you just one level of escalation. Like we put, just for example, the mRNA in between. Yeah. Still, this can be analyzed in the steady state and so on, so this is simple enough. And interestingly, and I think this is quite fun to notice, is actually that the steady state is now actually the product of these two sequential processes, of the one generating the mRNA and the other generating the protein. Yeah? So this is quite fun, and you can think, though, of course, that this generalizes. Yeah, is this already the most complex model of this transcription translation process? No, unfortunately not. Yeah? We will see actually a bit more complex models, but you can really go crazy here, like with bursty behavior, with noise, and so on. And like we will see a bit of this in the future. Okay. So what about regulation? I mean, up until now, our circuits are not very interesting. They just produce something, and that's it. And this is not very expressive, of course, because everybody at the end would be at the steady state, and that's it. Yeah? So how to actually regulate, control, remove stuff, and so on. And one simple way of regulation is actually repression. So this is schematically shown here. So we have a repressor molecule that can actually, in this case, bind to a certain binding site. Yeah? Like here shown as the domain D. And once it is bound, then this gene X cannot be anymore expressed. It goes to this state. So what is the rate by which it goes to the state? It's actually this constant times how many times you have a D, like this gene with a binding site, and how many times you have an R in your system. Yeah? So it's proportional to both concentrations. Yeah? And again, this is part of this linearity law that we saw before, that actually if you increase the concentration, the rate by which these things interact increases linearly with this. We will see a bit about this in the next lecture, why this is actually plausible. Yeah? But this is, if you think about it, first it sounds very simple, yeah? But then if you do the math, it's actually not so simple. Yeah? And it was, I think it was something that Einstein looked into, for example, with this ideal gas bumping together and so on. Yeah? Exactly, so here we have these two reactions here. And what we can be interested in now is actually how much of the time we have actually this occurrence here. Because whenever it's in this state, it's expressing something, and whenever this system is in this state, it's not expressing something. Yeah? So we are all interested in what is the concentration of averaged over time in the steady state, what is the concentration of Ds versus the complete D totals, yeah, that were in the system. And you can do the math. It's a bit longer. And there is a very important decision to do when you do the math. And some days ago, we had only one of these cases until we reviewed the lecture and added the other case. So what many researchers do is actually, they only consider this case 1 when they analyze the system. Yeah? And this is pretty plausible in many cases, but in some cases not, so we wanted to be precise in this lecture. Yeah? So what is the assumption? The assumption is actually that we have either 0, like absence of a repressor R, yeah? I mean, of course, the equation should work for this case, or we have actually very many of the repressors. Yeah? I mean, this is pretty plausible in many cases. I mean, I encourage you to do the research on how many repressor molecules you have in an E. coli cell, and you will see the answer is it depends on the repressor. But there exist repressors where you have many repressor molecules, and there exist repressors where you have only a few. And for the case where you have many, this analysis works out very nicely, and you get a very simple formula. And the answer is this here, this one here. So this is like a very nice Michaelis-Menten type formula. And if you don't do this simplification, you can also compute it, but it's a bit more involved, and you get actually this. So this is much less nicer, but this is an exact without any assumptions. And now that we have these two equations, we can actually plot it just to analyze it. And here you see actually what is happening. Yeah. So what is the what is the transcription translation rate of this whole system? So what is the proportion of the system being in the state where it's actually doing transcription translation, yeah, expression, yeah? And we see, of course, on the x-axis here, we see the repressor count. So if you have 0 repressors, it's doing this with a normalized rate of 1, yeah? So 100% is in the state where it's doing expression. And if you keep increasing the repressors, it's going down, 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 down. And of course, it would be tending towards 0, yeah? If everything is fully flooded with repressors, it's not doing anything anymore. Yeah? And I think what is interesting is here, this is the simplified formula, the solid line, yeah? So the very simple monoterm, and this is the exact formula. So you already see it works very well when there is 0, and it works very well when there is many. It's not doing so great of a job if it's in between. Yeah? Yeah? But most of the time, researchers just take this solid line because it's super simple to calculate with. But whenever you do that, be very careful and be aware that you do that. Yeah? And what I encourage you to do is to play a bit here with the numbers. Because if you play with the numbers, you will actually see cases where it would be a very bad idea to use the approximation. Okay, are there any questions until now, or is everything so far clear? Yeah?
Please let me know if I'm wrong, but till this moment, the efficiency of repressors are just considered based on the number of them.
Yeah.
Nothing else.
Yeah, completely, yeah. Mm-hmm. So you can, so for example, let me check where this is. I mean, you could tune the efficiency by actually tuning these parameters here. So this is already in the model, how easily they find their target and how strongly they bind, or how quickly they fall off the target again. Yeah. So this is the two kinetic parameters we have. Yeah. But that's for the moment everything we have. Yeah. Yeah. Mm-hmm.
So for the moment, the K is set to how long we are considering that they are sticking, or they are?
Okay, I think in this example I just set it to 1.
Okay.
Yeah, so here it was,
And
here the
one-
KD was just set to 1. Yeah.
One second?
Yeah. So this calculation was done unitless, so you can think of it, for example, 1 second. Yeah. But of course, like here it was much more. The purpose was much more to show the type of the curve that you're expecting. Yeah. But of course, like this curve will depend a lot on, of course, on your real constants. Yeah. Good, definitely. Yeah. Mm-hmm. But the point is, like, they will always look like this. Yeah. In this simple model. Yeah. Mm-hmm. Yes, and so this is perhaps the next complication of the model. You could think of leaky transcription and translation. I mean, this is very much the case for almost all repressors that are really around, yeah, and that you have to really consider when you build circuits. That actually, when something is in this repressed mode, it is still a little bit with a reduced rate transcribing or translating. Yeah. So this is actually accommodated here by a simple offset value that just offsets this. Yeah. And often this is enough. Like this simple model is typically enough to characterize some parts.
There is a question from.
Okay, yeah. Wait, is increasing protein? Yeah. So exactly. So in. Wait, I'm going to this picture here again. For example, here we would increase it not by multiplying, let's say, this binding site. You could also do that, yeah. But then you have to do very careful about the math, what that means here, yeah. What would be the simple in this simple model? It would be just multiplying this completely genetic construct. Yeah? So you would have, for example, you could think of that this whole stuff is on the plasmid, yeah, and you have a copy number, I don't know, of 100 of this plasmid. So you have actually 100 of these D-promoter gene constructs. But you could, of course, think of other designs. You could, of course, say, I want to have, let's say, 2 blocking sites next to my promoter that bind this repressor and so on. Yeah? But the math here would be a bit different. Yeah? But it's also like a classical thing to analyze, so it's definitely analyzable and a good idea.
There is another question.
Yeah, okay. Yes, we are assuming one repressor, so one binding site for a repressor for one promoter, and actually only one of the repressors can bind these binding sites, then it's occupied. So we don't have multiple repressors, for example, binding to the same repression site or so on. Yeah. So, which is of course definitely interesting if you analyze realistic repressors, yeah. But in this case, we really think of a very simple one.
Okay, I don't think it's a question, but an answer from another student.
Mm-hmm. Yeah, so I completely agree. Yeah, so thanks for the comment. I think it's more a comment, or yeah. Cool. But thanks a lot for. Yeah, feel free to post things. And by the way, also feel free to post things that you think are useful for the others, like in the Discord forum, because like unfortunately, like whatever you post here, I mean it is perfectly fine for the class, yeah. But it will be gone after the Zoom. So if you have some comments that others could help, feel free to post them in Discord. Yeah. Yeah, so we can, I don't go into details, but we can do the same game for actuators, yeah? And again here, it's a very simple genetic construct. It has only one binding site, only one actuator can go to this one binding site, yeah? Yeah? But the model consists of, it considers that we have multiple of these constructs again. So this is perfectly possible. Yeah? This is just the number D in the model is actually the number of constructs that you have in your cell. Yeah? So which is considered to be, D total is considered to be a constant. Yeah? But you could vary it, of course. Yeah? Yeah, and not very surprisingly, the activator function looks exactly like the kind of opposite of the repressor function. I think we have them drawn at some place together. Okay, I thought so. It's not doing that. Okay, let me fix that later. But essentially, this is the activator function. That means like if you have 0 activators, you have 0 expression. And of course, when you keep increasing your activators, you're converging to 100% of the time the system being in the activated state. Yeah. And actually, it should plot here the activation function. I don't know what I messed up, but I will fix it after the class and I'll upload it. Okay, and this brings us to a final question. So we have activation, we have repression as the very simplest genetic constructs. Yeah? And actually in the problem session, you will look into a bit differences of this both. Yeah? Because it's not, and we will see this I think in the third lecture, we will look into this in more detail, because it's not very clear what is good for a cell in which context. Yeah? And there's very interesting answers to this. Yeah? But this is going to happen in a future class. Huh? And
another question.
Yeah. Okay.
Can you explain the graph significance again, please?
Okay.
I think it's the previous one.
This one here. Do you mean like this graph here? Okay. Yeah. So this was actually, so if perhaps I show it in relation to the repression curve. So that was our repression curve, right? So we had on the x-axis here, we had the concentration or the number of repressors in the system, and on the y-axis we had the relative concentrations of the system being in the expressing state. And of course, like when we have 0 repressors, 100% of the system is in the expressing state. And as we keep increasing the repressors, the system tends to 0% being in the expressing state. And this is to compare with this figure, so which is this genetic construct here. And it's doing exactly the opposite. Yeah? When you have 0 activators, yeah, actually 0% of your system is in the expressing state. And as you keep increasing the activators, for example, in this case to 20, yeah. So it's assuming like you have something like 20 molecules of activator here. Yeah? Your system tends to be 100% in the activating state. Yeah? Yeah?
To fix this graph, you need to define R. You forgot to define big R.
Okay, thanks a lot. Okay, thanks. Thank you very much. It was working yesterday, and I don't know, I fixed something. Thank you. Thanks.
Question.
Okay, thanks. Yeah. Mm-hmm.
Could you please give us an understanding, like how does the leakiness also with activators work? I mean,
You mean why it's actually here?
Yeah.
Okay.
I mean, how does the activator?
Yeah. So it depends a bit on of course, like here it's very abstract what this activator. It's just sitting there and then it works magically. Yeah? But often what this thing is doing actually is like stabilizing. So there is, for example, the RNA polymerase coming here, yeah, and trying to attach here to this promoter sequence. Yeah? And let's say this promoter sequence doesn't have a very good binding energy with the RNA polymerase. So the RNA, I mean, everything is a stochastic process. So this RNA polymerase comes and falls off relatively quickly. I mean, it sticks a bit, but it falls off relatively quickly. Yeah? But it could be actually that this activator sitting here next to it is actually interacting with this RNA polymerase to kind of pull it down to the strand. So whenever there is an activator sitting here, this RNA polymerase can really now grab the DNA strand and start transcribing. So this would be, for example, one mechanism. And why would it be leaky? Because even if it's not here, it has some probability of it working. It's a stochastic process. Yeah? I mean, one big thing, like all our models here were ODEs. Yeah? So you didn't see any kind of noise. Yeah? But we will see actually that everything of this CRNs is a purely stochastic process. Yeah? We just didn't want to throw all the problems at you at the first class, yeah? But like we will look into this, into the stochasticity of these processes, yeah? And that this is really a problem. Yeah, yeah? Thanks for the question. Was there? Yeah, exactly. Yeah, thanks for the question, and thank you for answering it. Actually, I think that's it. Yeah.
I think it's
Forwards?
another question.
It's another.
Yeah.
So one was actually to get the parameters k plus k minus. Yeah, exactly. I mean, there might be like if there is experts here in the forum, please go ahead and answer this. Like if you did find out k pluses and k minuses, yeah. But what we do is we take, so the first thing is like it's relatively easily to fit it to find the quotient between k minus divided by k plus, because this is actually the steady state, yeah. So you read this from the steady state. It's relatively difficult to get the real k plus and k minus, because the real k plus k minus, of course, this depends now on the timing. So you need to have time data, yeah. Or so. Yeah, essentially what you do is actually you look at timed experimental data and try to fit this. But this is not at all trivial. This is really complicated. Cool. Thanks a lot. Then so this brings me to the end of the first lecture part. And I think we are in time. Yeah, like 2 minutes before. Great. So how do we progress? Like the idea now is actually you will find some problems. If you go to the problem session here, and if you click on the first one being modeling, yeah. So we have compiled 3 problems for you. They are of different difficulty. But the difficulty depends a bit on your background. So for some people, 3 might be very long. For some, 3 is very quick if you did parameter fitting before. 3 is actually, it's yeah. So 1 and 2 is a bit the understanding of this lecture, yeah. And 3 is a bit fitting on real bioproduction data to create this very simple model and to fit it automatically. And yeah, what we do now is like we will stop actually the streaming for the moment. We will be here in class, and we of course read your messages. So keep free, like if you're present, like keep free poking us and or chat online. Yeah. And we will come back after some time. So Mariapia, do you know?
Maybe a couple of hours.
Yeah. So I think something like 2 hours. But let me check this in the.
Yes.
Yeah, we have written it actually in the Discord forum, I think. Let me go to the very beginning. Yeah, so at 4:30 we come back, and then we will quickly discuss. We will quickly discuss solutions. We will not really present all the solutions, but we will just give some hints and discuss what you have came up. Yeah, and thanks a lot for joining for this first lecture. And yeah, of course, like by the way, everything will be online. So since it's the first lecture and it's our first time recording stuff, I don't know how long it will take us to put it online, but hopefully not too long. Yeah, but once it's online, we will announce it, of course. Yeah. And yes, thanks a lot for coming, and hope to see you afterwards. Yeah, bye.